हेलो फ्रेंड्स, ए वेरी गुड मॉर्निंग टू ऑल ऑफ यू। आज मैं आप लोगों के लिए एक नया वीडियो ब्रेन बायोकेमिस्ट्री ड्यूरिंग स्टारवेशन प्रोलॉन्ग्ड स्टारवेशन लेकर आया हूं। आप इस वीडियो को अंत तक देखना ना भूलेंगे। आशा करता हूं ये वीडियो आपको जरूर पसंद आएगा। अगर आपने अभी तक मेरे YouTube चैनल को सब्सक्राइब नहीं किया हो तो आप इसे जरूर सब्सक्राइब कर लेंगे। तो चलिए वीडियो की शुरुआत करते हैं। जैसा कि हम लोग जानते हैं, द ब्रेन इज द मोस्ट कॉम्प्लेक्स ऑर्गन इन द ह्यूमन बॉडी। इट कंटेन्स बिलियंस ऑफ नर्व फाइबर, axon and dendrites. The brain makes up about 2% of human body weight, but it utilizes approximately 20% of the body energy. The objective of this video is to develop idea about ketone body's metabolism, how ketone body's metabolism takes place in depletion of glucose. Second one, to understand brain biochemistry during starvation, to develop an idea that how hypoglycemia is linked with brain starvation, to explore the way that how brain utilizes ketone bodies as source of energy during starvation or prolonged starvation condition. First of all, what is starvation? Starvation is a severe deficiency in caloric energy intake below the level needed to maintain an organism life. Ketone bodies are utilized for energy needs in the extrahepatic tissue such as brain, heart, skeletal muscle and kidney. Ketone bodies are not oxidized in the liver because the enzymes succinyl coenzyme A, acetoacetate coenzyme transferase, thiophrase is not expressed in the liver. Hypoglycemia is brain starvation. In such conditions, the concentration of glucose is decreases. In such condition, brain first recognize the problem, resulting in confusion, lightheadedness, a loss of balance, slurred speech, and impaired vision. This state is called hypoglycemia or low blood glucose. Hypoglycemia can be very dangerous and sometimes fatal. That's why it is very important to ensure that you recognize the symptom of hypoglycemia immediately and consume carbohydrate-rich food to restore your brain function back to normal once again. But in case of a starvation condition, what happened? Since brain lacks the biological mechanism necessary to burn amino acid and fatty acid, so it depends on glucose for the bulk of its energy demand. But when glucose level is depleted, as for example in extreme starvation, in such condition, fat is metabolized through beta oxidation to compensate the energy demand. Beta oxidation of fatty acid leads to production of ketone bodies. Synthesis of ketone bodies is known as ketogenesis. There are three types of ketone bodies: acetoacetate, beta hydroxybutyrate, and acetone. Beta hydroxy beta methylglycerol coenzyme A or HMG coenzyme A, in presence of enzyme HMG coenzyme A lyase, it splitted its acyl coenzyme A and converted to acetoacetate. Acetoacetate is non-enzymatic decarboxylation takes place in acetoacetate and it is converted into acetone. The reaction is decarboxylation reaction. Acetoacetate is converted into beta hydroxybutyrate. It is reversible reaction. Since ketone body can cross blood-brain barrier, so it can be easily metabolized in brain easily. Blood-brain barrier don't allow to pass fatty acid directly into brain during shortage of glucose. So first, fat is metabolized, fat is burned in absence of carbohydrate. If fat is metabolized through beta oxidation and leads to production of ketone bodies. Ketone bodies metabolism. Primary source of energy is glucose, but when the glucose level is depleted, then glycogen is utilized. From liver and muscle cells. But when the level of glycogen is depleted, then fat is metabolized through beta oxidation. First of all, fatty acid is converted to fatty acyl coenzyme A. In mitochondria of liver cell, fatty acyl coenzyme A, there is beta oxidation takes place to form acyl coenzyme A. Acetyl coenzyme A, in presence of enzyme HMG coenzyme A synthase, it is converted into HMG coenzyme A. In presence of enzyme HMG coenzyme A lyase, it is converted to acetoacetate. Acetoacetate is directly converted into hydroxybutyrate. Acetoacetate go to the circulation and it is finally go to peripheral tissue. In peripheral tissue, acetoacetate in presence of enzyme thiophorase, it is converted into acetoacetyl coenzyme A. Acetoacetyl coenzyme A further converted into acetyl coenzyme A and provide energy to the brain. The production of ketone bodies takes place inside liver. These ketone bodies go to release into the circulation and finally these ketones are metabolized by peripheral tissues as for example muscles, renal cortex, brain during starvation. Then what is the chemistry of brain during a starvation condition? This diagram shows first of all we show brain in fasting condition. Brain in fasting condition, it produces ketone bodies as for example acetoacetate, beta hydroxybutyrate, ketone bodies is produced during a starvation condition because it is used by brain to meet energy need. Brain biochemistry during starvation. What happened when glucose is depleted? The level of glucose is depleted. Then what happened? If the level of glucose decreased, then production of ketone bodies from beta oxidation. Ketone bodies goes to peripheral tissue as for example brain and here it is converted into acetyl coenzyme A and it goes to TCA cycle to generate energy and this is the energy source during a starvation condition. But in case of well-fed condition, glucose is the source of energy. Glucose directly goes to, it crosses the blood-brain barrier and goes to inside brain, and it is the ultimate source during well-fed condition, and it is converted into acetyl coenzyme A. This acetyl coenzyme A goes to TCA cycle to produce energy. But in case of starvation, ketone bodies is the source of acetyl coenzyme A inside the brain, which further go to TCA cycle. In case of prolonged starvation, first priority is to provide sufficient glucose to brain and other tissue that are dependent on it. Second priority, preserve protein. Shift from utilization of glucose to utilization of fatty acid plus ketone bodies. In case of glucose is blood glucose level depleted, in case of prolonged, as for example, in case of prolonged starvation, then there is shift from utilization of glucose to utilization of fatty acid plus ketone bodies. Then fat is metabolized through beta oxidation to compensate the energy demand. Oxidation of fatty acid leads to production of ketone bodies. Mobilization of TAG in adequate tissue, gluconeogenesis by liver, muscle shift from glucose to fatty acid as fuel. After 3 days of starvation, liver forms large amount of ketone bodies. Why ketone bodies is produced in large amount? This is due to shortage of oxaloacetate. Oxaloacetate is intermediary product of TCA cycle. There is deficiency of oxaloacetate takes place. This is due to oxaloacetate is converted its part. It is utilized in gluconeogenesis process. But beta oxidation leads to production of acetyl coenzyme A during prolonged starvation condition. Hence, acetyl coenzyme A pool is increases during such condition because oxaloacetate is not available for condensation with acetyl coenzyme A, because oxaloacetate is utilized in gluconeogenesis process. Hence, TCA cycle is hampered. Released into blood, brain and heart start to use ketone bodies as fuel source. After several weeks of starvation, ketone bodies major fuel of brain. This can be depicted in this picture. After depletion of tag stores, proteins degradation accelerates. And finally, death due to loss of heart, liver, and kidney function. This is the overall mechanism during prolonged starvation and starvation condition. In such condition, ketone body is the source of energy. In nutshell, in case of starvation, exhaust of glycogen from muscles and liver, use of stored lipids. In absence of glucose, fat is burned through beta oxidation to produce ketone bodies, and this ketone bodies is further utilized by brain as a source of energy. तो इस वीडियो में हम लोगों ने जाना कि किस प्रकार से कीटोन बॉडीज ब्रेन के लिए सोर्स ऑफ एनर्जी का काम करता है ड्यूरिंग स्टारवेशन और प्रोलॉन्ग्ड स्टारवेशन कंडीशन में। स्टारवेशन कंडीशन में ग्लूकोज का लेवल का कम होना फैट के मेटाबॉलिज्म को बढ़ाता है, जिससे कीटोन बॉडी सिंथेसिस कीटोजेनेसिस प्रोसेस प्रारंभ होती है। फर्दर ये कीटोन बॉडीज को ब्रेन सोर्स ऑफ एनर्जी के रूप में इस्तेमाल करता है। थैंक यू। अगर आपको वीडियो पसंद आया होगा तो आप इसे शेयर करना ना भूलेंगे।